Okay, good morning, afternoon, and evening, everyone. Welcome to the 18th session on of the GO2NE Global Ocean Oxygen Network webinar series. My name is Sean Crowe from the Departments of Microbiology and Immunology and Earth, Ocean, and Atmospheric Sciences at the University of British Columbia in Vancouver, Canada. The whole GO2NE team is very happy that you took the time to be with us and our incredible speakers this morning, afternoon, and evening. Before starting, we'd like to bring your attention to the recently launched ocean deoxygenation newsletter, Good News, which reports on the latest activities of the GO2NE group. The QR code will take you to the most recent issue. Finally, the next GO2NE webinar has yet to be scheduled, but is planned for March 2023. So please look out for details to register when the webinar is announced. Furthermore, here are some housekeeping announcements. You as attendees have the possibility to type your questions into the question box. The chat can be used to share additional information if you have any logistical issues. So let's get started. Today we have two wonderful scientists speaking. We will start with Kohen Bauer, who's from Ocean Networks Canada at the University of Victoria here in Canada, followed by Dan Mills, who's based in the Department of Earth and Environmental Sciences, Paleontology and Geobiology at the University of Munich. Both speakers will present for 20 minutes, followed by a 10-minute question and answer period. So I'll get started by introducing Dr. Kohen Bauer. Dr. Bauer got his undergraduate degree in geology from Queen's University in Canada, then moved on to do a master's degree in geological sciences at the University of Toronto. And after this, he came to Vancouver to do a PhD at the University of British Columbia. After that, he moved on to a postdoctoral position at the University of Hong Kong, and he is currently a senior scientist at Ocean Networks Canada, where he's doing a variety of things, including developing and deploying sensors for ocean oxygen monitoring. So I'll turn the floor over to Kohen.
All right, thank you very much. I'm going to share my screen now. So is everyone able to see my screen okay? All right. Thank you very much, Sean, for the introduction, and thank you for the organizers of the webinar for the opportunity to share some of my work today. I really appreciate the opportunity. Today, I'm going to be speaking to you about ocean anoxia in deep time, and in particular, an ocean deoxygenation episode that occurred during the Cretaceous. Firstly, I'd just like to start by acknowledging where we got some money to perform some of this research. So without this funding, a lot of this work wouldn't have been possible. So thank you for that. I'd also like to give a major shout out to my fantastic colleagues all over the world that participated in aspects of this research. Collaborating with them is extremely fun and makes doing this work really, really enjoyable. So I have to give a shout out to them. All right, so it wouldn't be a GO2NE webinar without showing this figure from Denise Breitberg's 2018 science paper. Really, what this is aiming to illustrate is the rates and spatial extent of modern ocean deoxygenation, right? And while we're aware that this ocean deoxygenation has numerous negative effects on biological and biogeochemical processes, we still don't have a full understanding of what the impacts of ocean deoxygenation might be on the Earth system, in particular to climate and biology. Furthermore, we don't know if these impacts are really tethered or dependent on the rates and spatial magnitude of anoxia or ocean deoxygenation, low oxygen conditions in the oceans. Furthermore, this is a very modern issue. But we know that oxygen contents in the ocean have fluctuated greatly over Earth's history. And this really informs us as to how or what the impacts may be with ocean deoxygenation going forward. And so I want to take a step back and provide a little geologic perspective on the redox evolution of the Earth system through time. And really, it's this redox evolution of the ocean atmosphere system that is intimately linked to the evolution of climate and biology across the geologic record. So what I'm showing here is 4 billion years of Earth's history on the x-axis, and concentration of different or the major electron acceptors in seawater, right? And so if we take oxygen, for example, we can see that oxygen concentrations in the ocean have changed greatly over geologic time, from nearly nothing in the Archean through to modern-like concentrations at the modern day, right? Importantly, it's these fluctuations or major changes in the oxygen concentration of seawater that are contemporaneous or associated with major climatic events in Earth's history, such as some of the Earth's most serious glaciations. Likewise, changes or fluctuations in the ocean redox state or ocean oxygen concentrations are linked to some of the most important biological events in the history of life on Earth. The emergence of the first life through to the great oxidation, where we see maybe the emergence of multicellular life, eukaryotes. Dan is going to provide some great information on this in the second half of the webinar. And then, you know, at the beginning of the Phanerozoic eon, we have a change to modern-like ocean oxygen concentrations. Moving forward in time again, we're in the Cenozoic era, or the last 65 million years of Earth's history, where we have climate dynamics on glacial, interglacial timescales, and again, associated changes in ocean oxygen concentrations. We take one step forward again, and we're in the so-called human epoch, or the Anthropocene. And now we as humans are acting as biogeochemical agents in the Earth system, changing things like climate and ocean oxygen content. And again, there's going to be associated feedbacks and impacts on the Earth system associated with us changing the Earth life climate system. And so really we come full circle, right, back to this modern day picture. And from our geologic perspective, we know that ocean deoxygenation, or changing ocean oxygen concentrations, can drive a cascade of biogeochemical and climatic effects in the Earth system that ultimately have really important impacts for the evolution of life and climate. And it's really going into the geologic record and having this geologic perspective that provides a window as to how the Earth system will respond going forward in the face of modern ocean deoxygenation. And that really sets up my motivation, what I do as a researcher, what I work on, and it's really, I aim to create robust models of biogeochemical cycles that describe and predict the response of the Earth life climate system during times of biological, climatic, and geochemical change across both geologic and human timescales. And I would argue that, again, this would allow us to improve our capacity to predict and respond to modern change that is driven by humans. Okay, so what do I actually do? Well, mainly I study the Phanerozoic rock record, right? So the Phanerozoic eon is the last 550 million years of Earth's history. And during most of this time interval, we have modern-like ocean atmosphere concentrations, right? So oxygen concentrations in the ocean are much as they are today. However, there's key exceptions. And these key exceptions are known as oceanic anoxic events. And these are intervals or short intervals in Earth's history where we believe the oceans developed widespread anoxia on sort of shorter timescales, right? These events, importantly, are associated with some of the greatest perturbations to life and climate and biogeochemical cycles across this time interval. And so really, these events, these ocean deoxygenation events, provide very important analogs or ancient deoxygenation analogs through which we can examine the response of the Earth system in the face of changing ocean oxygen concentrations. However, many of the aspects of these events are still poorly constrained, and in particular, estimates for the rates and spatial extent of ocean deoxygenation across these different events are still largely unknown. And so in particular, I study OAE-1A. It's an ocean deoxygenation event or oceanic anoxic event that occurred 120 million years in the Cretaceous. And so what is an OAE in the geologic record? How does an event like this manifest? Well, in particular, they can be quite conspicuous, actually, and they're commonly associated with the deposition of organic matter-rich marine sediment that implies the deposition under oxygen poor conditions. So you can imagine you have typical or well-oxygenated background marine sedimentation. These are carbonates that are eventually lithified into limestones. We have an environmental perturbation, which the oceans become anoxic, and we have the deposition of organic matter-rich marine sediments. This implies deposition under oxygen-poor conditions, as I mentioned. And then the recovery from the event is commonly associated with the return to normal marine or background-like sedimentation, right? I have to remind the audience that the Earth system 120 million years ago looked very different than it does today, right? So we had a different paleogeography. In particular, 120 million years ago, the continents were in a different arrangement. And so we had two main oceans, the Tethys Ocean, which is this ocean here, and then a very large Pacific, or also known as the Paleontologic Ocean. And to study these events, or to study OAE1A 120 million years ago, we need to examine rocks or sediments that were deposited in the oceans at this time, right? And so I work on samples that were deposited in the Western Tethys Ocean, known as the Chisman drill core. And I also have samples from DSTP Site 463, and these sediments were originally laid down in the Pacific or Paleontologic Ocean. This is another paleogeographic reconstruction of where we have our samples from. So to test redox conditions during OAE1A, again, I study samples deposited in the Western Tethys Ocean, known as the Chisman drill core, which are now rocks uplifted in Italy. Similarly, I also have sediments from DSTP Site 463. These sediments were originally deposited in the Pacific or Paleontologic Ocean. They exist there today, except now they're buried in their rocks on the seafloor, and they're accessed via the Integrated Ocean Discovery Program. Okay, so what does the anatomy of OAE1A look like? So what I'm showing here is data or geochemical and lithological information from the Tethys Ocean, or the Chisman core, and the Pacific Ocean, or DSTP Site 463 on the right. I want to start by bringing your attention to this stratigraphic log, right? So this is as I mentioned previously, the deposition of carbonates before the event, which then transitioned to this very organic matter-rich black shale lithology. It's colloquially referred to as a black shale. And this really represents the body of OAE1A. And then after the event, we transition back to carbonate sedimentation. You can see that the increase in organic carbon contents increases during this interval, and this lasts about 1 million years. So how do we actually know that the oceans went anoxic across this time scale? Well, we use geochemical indicators, geochemical proxies, or paleoredox proxies. And these are geochemical tools which allow us to diagnose sediment deposition under anoxic conditions. And there's many of them. And really, the story that they tell for both sites is that indeed the oceans develop pervasive anoxia at both locations across this entire event. Similarly, the story is very much the same for the Pacific Ocean site, where we have an increase in organic matter deposition during the event, as well as geochemical and paleoredox proxies telling us that the oceans developed anoxia. So what are some more nuanced questions that we can ask about this event, and what can we learn about the Earth system in the face of deoxygenation? Well, the first question we want to ask is, was deoxygenation global? And I sort of gave away the punchline for that question already, but I think more importantly, a key question that we should ask is, what were the rates of ocean deoxygenation associated with this particular event in Earth's history? Were they slow? Were they fast? And to really answer these two questions, we establish multiple geochemical records in these same cores, okay? And so I'm going to walk you through this. The Chisman core, again on the left, the Tethys Ocean, and DSTP Site 463 on the right, the Pacific Ocean. The OAE1A interval is in green here. This is its duration, as well as here in the Pacific Ocean. Pre-OAE1A is on the right, and of course the green shading represents the event. All of these panels represent different paleoredox or geochemical proxies. And how they work actually is if they fall within this pink shading, it means that we have normal or oxygenated background conditions. And once the proxies transition out of this pink shading into the green shading, we can confidently assess or interpret sediment deposition under anoxic conditions, right? And so really in the Chisman core, this transition out of background conditions to the event itself happens really between two data points in this sample resolution. And we know that anoxia is established here. And so this deoxygenation interval based on the time scale is about 30,000 years. So that's pretty rapid. Likewise, the story is very much the same in the Pacific Ocean. We have a transition at the beginning of OAE1A that is coval in all of these proxies that we can confidently diagnose deposition under anoxic conditions. And anoxia was actually established at almost precisely the same time at both locations and over very rapid timescales. And so really the conclusion of this first piece of work were that large swaths of the oceans became anoxic from the Tethys to the Pacific in less than 30,000 years. Now this is pretty rapid. However, it's also very much comparable to other oceanic anoxic events that developed during the Cretaceous. Another take-home message from this work was that once we established anoxia in the ocean, it was very much persistent. So we do not see a return to oxygenated conditions at either location. And so this is implying that anoxia was existing for about 1 million years after it developed very rapidly. Okay, so can we actually do even better? Can we obtain even more precise estimates for the rates and the spatial magnitude of anoxia in the Earth system at this time? And we wanted to go a step further. And really to do this, we needed much more high resolution geological and geochemical records. And so we went back to the Chisholm core in a new set of work that we're producing right now. Again, this is the same Chisholm core in the Tethys Ocean that I showed you before, except now my colleagues resampled this core at centimeter scale resolution, right? And so this is about 20 meters of core. What this does is it produces a lot more samples. There's now 230 samples, and it yields a complete 3 million year snapshot of Earth's history through the early Cretaceous during this time interval. And these samples now yield geochemical or environmental information on millennial scale resolution, right? So now we can really start to address questions on much more refined and high resolution timescales, which is really important. What do we actually learn from this? Well, here's sort of what we're finding, and I'll walk you through this. The top panels, again, are paleoredox proxies, and these orange panels are paleoclimate proxies. I'm not going to go into detail today about what these proxies can tell us. However, similar to the last panels, pre-OAE1A is on the right. The OAE1A event is shaded in green here, and then we have the post-event occurring after the green shading. These paleoredox proxies, again, simply put, if you see blue shading, it means that they've crossed their threshold and we can interpret or diagnose sediment deposition under oxygen poor conditions. I think the first message here from this work is that the transition from oxic to anoxic conditions during the event is extremely rapid, right? You can now see that this transition is very stark. Based on the timescales, now we can say that it is potentially less than 10,000 years, right? And so now this is an order of magnitude faster than that our previous work showed us. This is remarkably rapid. Another message here is that you can see during the event, we have persistently anoxic conditions in all of these proxies, right? And so the message is that, or the interpretation is that anoxia was really persistent across a 1 million year timescale. We're actually learning some very exciting and new things with this high resolution data set that was not previously observed. I think the first take home message is that reoxygenation of the oceans after this event was also very rapid on the time scale of tens of thousands of years. However, a new observation, which was really cool, is that after the main persistent OAE, after the event, the Earth system sort of drops into this oscillatory phase where we have a cyclical transition from oxygenated conditions to anoxic conditions that occur on these Milankovitch or 100,000 or 100,000 kiloyear, 100,000 year timescales. So this is quite remarkable. These are almost mini OAEs that happen after a persistent OAE existed for about 1 million years. Again, all of these transitions are coval in our proxies, and if you eyeball it, you can see that they are also sort of in sync with these climate-related proxies. And so really the two main messages here is that based on our high-resolution data set, we really can drill down into these refined timescales, and that ocean deoxygenation and reoxygenation is occurring on very rapid timescales, approaching human-like timescales, I would argue, and possibly related to, orbital or Milankovitch-like climate forcing. I'll also just take a step back, and if you look at this 3 million year interval in Earth's history in general, ocean oxygen dynamics really characterize this interval in Earth's history. So we can see that we shifted from, anoxic persistent conditions to this cyclatory or oscillatory phase after. And so, changing ocean oxygen conditions really characterize this interval in Earth's history. The next aspect of this work is that we wanted to constrain the spatial extent of ocean anoxia during these intervals. And to do this, we constructed a simple mass balance model based on cerium anomalies. And simply how this works is a 2-sync mass balance model. To drive a positive cerium anomaly shift in the OAE1 sediments, which is what we observe, we simply have to dial back or reduce the size of the oxic sink for cerium in the oceans. And this happens to be manganese oxides. And so when we do this in our model, to drive the observed shift in the OAE1A sediments, we need to increase ocean anoxia to about 10 to 25% during the event itself. And so now through time, we have a sort of a quantitative estimate for how much seafloor anoxia we had during the event. And so this is about 15 to 20%. This is comparable to other oceanic anoxic events that occurred during the Cretaceous. But I think notably, and perhaps a little remarkably, our numerical models are also showing that during these oscillatory or mini transient OAEs that happen after this persistent OAE, the Earth system also had appreciable anoxia, seafloor anoxia, somewhere between 5 to 10%. So perhaps this is quite surprising that even during these transient OAEs, we're developing, quite large swaths of the oceans were anoxic and on very rapid timescales. So this summarizes the findings of this work so far. The numerical models are estimating appreciable ocean anoxia during both transient and persistent anoxic events, and that this is implying that severe levels of ocean deoxygenation on rapid timescales may be related to a warming world and climate forcing in the Earth system. Again, taking a final step back in the interest of time, I see I have about 30 seconds left. I just want to bring us back to this perspective that these OAEs really represent windows or analogs of ocean deoxygenation in Earth's history, where we see biological crises, climate crises, and they provide analogs as to which we can learn something about the Earth system, the response of the Earth system to ocean deoxygenation that is currently impacting the modern oceans. And again, I think there's lots of future directions, things that we can learn going forward. We're going to continue to refine our geochemical proxies or paleoredox proxies that allow us to make robust interpretations of the geologic record. I would argue that we really should focus on collecting high-resolution records. If we want to address processes on very high resolution or short timescales, we need records that actually capture that information. We should develop models or computer models that are directly tethered to geochemical data, and we can continuously refine our frameworks for what the real feedbacks associated with ocean deoxygenation in deep time as well as the modern might be. So thank you very much for listening, and now I'll try and field any questions that you may have.
Okay, thanks very much, Kohen, for a very interesting talk. So I see we have a number of questions that have come from the audience. So can you see those, or shall I read them out to you?
If you could organize them for me, that would be very helpful.
Yeah. Okay, so the first question is, what were the approximate paleo water depths of the 2 core sites?
Yeah, fantastic question. So the Chisman or the Western Tethys was I could go back to this paleo map here. So the Western Tethys, which is where you see the Chisman core in the top right, potentially about 1,000 to 1,500 meters of water depth is the Chisman core deposition, and as well as DSTP site 463, it was a more open ocean site, and the paleo water depths were maybe more poorly constrained, but between 1,500 and possibly 2,000 meters. Yeah. So, but very similar.
Okay, thank you. So the next question is how can the high temporal variability seen between 119 and 118 MA be interpreted? I think
119.
this is referring to the high
This is
resolution.
for these oscillatory phases.
I think so.
Yeah, this is a great question. This is part of what we're trying to figure out now. You know, I think it's related to climate forcing. As you can see in this paleoclimate reconstructions based on some weathering proxies. I said in the talk that this is Milankovitch-like timescales or astronomical forcing-like timescales. I didn't want to say explicitly that this is related to precession or eccentricity because I haven't quantitatively done the cyclostratigraphy or the time series analysis that would allow me to robustly tell you that this is related to some sort of astronomical parameter. I will also say that the age model that I'm displaying here is currently being refined. So this is subject to change. It's not going to change the interpretations. The points will shift relative to one another, but the message will remain the same. And so my colleagues in Italy are currently working on a refined timescale here. However, yes, if you do the eyeball test, you can really see some cycles in here. And I would argue that this is most likely linked to astronomical or Milankovitch-like cycles. The interesting question then I think that extends from this is that, so why do we have this sort of blanket or persistent OAE and that these cycles only emerge after, right? So is the Earth system, so what I also didn't mention at the beginning of the talk is that there was a large igneous province eruption prior to this event. So potentially a very large injection of CO2 into the atmosphere associated with the onset of this event. So perhaps the Earth system is trying to mop up this CO2 during the body of OAE-1A, and then it finally is able to recover, but there's still sort of enough climate forcing left in the system, so perhaps some sort of hysteresis that allows you to drop into this oscillatory-like phase. But it's really, I think the observation is quite interesting that we have, you know, pervasive anoxia redevelop on very rapid timescales. So I hope that answered your question.
Super, thanks. So we've got lots of questions coming up here. We've got about 5 minutes or so, 4 or 5 minutes left to answer them. So the next question is your redox proxies show interesting differences, for instance, consistent manganese depletion, but highly variable iron enrichment during the OAE. Is this telling you that degree of anoxia was in fact highly variable during the event, or do you interpret it differently?
Yeah, that's a good question. There's a couple things that we need to think about there. So obviously these redox proxies have different residence times in seawater. So there might be the variability I would most likely attribute to how quickly they are supplied and removed from the ocean environment, especially given pervasive anoxia. I think the main observation is that almost all of them are above the conservative thresholds, I would argue, right? So even though there is some cyclicity during the body of the OEE itself, the main message is that they all tell you that there was widespread anoxia. So yeah, I think that the differences during the body are most likely related to supply and removal and then the residence time of these elements in seawater.
Okay. So the next question is, what was the oxygen concentration during anoxia in milligrams per liter? And what was the ocean temperature at this time? And was this only in the tropics?
Yeah, lots of good questions there. Really, what these proxies would tell you is potentially zero, right? A deposition under anoxic conditions, definitely at this location. It's really hard to gain sort of quantitative information about a precise oxygen estimate, right? It's really almost a binary like signal. The temperature of the oceans was much warmer at this time, the Cretaceous, and certainly the early Cretaceous was a greenhouse climate, and so we had, you know, much warmer ocean temperatures than we do today. And yes, these cores come from higher latitudes, And so, but we see similar signals in, OE1A is can be correlated to sites globally, not just only in the tropics or at the mid latitudes. We really see this event occur in our records globally. So it's not, it was across all latitudes, I would argue.
Okay, so the next question is, thanks for the excellent talk. You showed atmospheric PO2 over the last 500 million years. How tightly is this correlated with the ocean oxygen inventory on these timescales?
Yeah, it's pretty difficult to see, right? So this is, if I go back, this was, yeah, right. So if you look at this sort of reconstruction, it's really difficult to tie in atmospheric oxygen concentrations to ocean concentrations. I think that we're getting there. I think with higher resolution data sets and with some proxies that are in development that can really tell you about atmospheric oxygen concentrations, that there might be a link there. And it would be quite remarkable to make a discovery about changing atmospheric oxygen concentrations across these, 3 million year, 1 million year like timescales. So I think if you can make that connection, which many are working on today in the community, that would be a remarkable finding. But it's really difficult across this 3 million year chunk of Earth's history to tie it to this very high level curve. So that's what I can say for now.
Okay, thanks. So we still have lots of great questions, but we're out of time, so we're going to have to move on to the next speaker. The questions are recorded, and so Dr. Bauer will address these after the webinar. Thank you again, Kohen.
Yeah, thank you very much, everyone, for listening. I appreciate it. And I'm going to stop sharing my screen now.
Okay, so our next speaker is Dr. Dan Mills, who is a research associate at the University of Munich. Dan did his undergrad degree in geology at Penn State University. He then went on to do a PhD in the Nordic Center for Earth Evolution in Denmark. And after that, moved on to a postdoc at Stanford before transitioning to his current position in Munich. So Dan's going to continue our discussion of oxygen in the past and shift focus now to its effects on biology. So I'll turn the floor over to you, Dan.
All right, well, thank you very much. Thanks for the introduction, Sean. Kohen, thanks for the great talk. It sets up mine very nicely. And thanks to the organizers for giving me this opportunity to share my research with everybody. So first of all, this research was done when I was a postdoc funded by the Agaron Institute, and I was hosted by Eric Sperling at Stanford. And then this work was done in collaboration with my colleagues at UBC in Vancouver, and then as well as my colleague at Hui. So these are the people who all came together to present this work. All right, so I'm going to be talking about eukaryotes. This thing here is not a eukaryote. This is your garden variety flagellated bacteria. These are the things that we're not going to be talking about. We're going to be talking about things like this. So this is your garden variety alga. So this is a eukaryotic organism. You know this because its DNA is contained in a membrane-bound nucleus, and it has these intracellular organelles, these metabolic organelles like mitochondria, where aerobic respiration takes place in this case, and then as a chloroplast, where photosynthesis takes place. So we're talking about eukaryotes like this, and going to ignore bacteria and archaea for this talk. And then, of course, we're going to look at how the ecology of microbial eukaryotes shifts as a function of oxygen availability. So here's this figure, this drawing from Tom Fenchel and Bland Finlay, where it's just showing the relative energy flow in a typical marine ecosystem that is fully oxygenated by modern standards. And aerobic respiration compared to fermentation is sufficiently efficient to allow for multiple trophic levels, because there's enough energy left over after each trophic level to support an additional one. And in this case, everything you see here is a eukaryote. Under anoxia, ecosystems or food chains look more like this, where you do have predatory protists, microbial eukaryotes that are eating bacteria. But that's essentially it. There's not enough energy left over in the scheme to support larger eukaryotes like animals, and then larger animals like large fish. So this is essentially what eukaryote ecosystems and ecosystems in general look like under anoxia. So the origin of eukaryotes haven't always existed on Earth. So the origin of eukaryotes was really, in a sense, the origin of food webs as we understand them now. Because again, everything pictured here is a eukaryote. And bacteria and archaea, while some bacteria may prey on each other, they don't engulf one another. They can't phagocytose. They can't swallow other cells, and food webs in strict sense don't really exist. So the origin of eukaryotes really is the origin of modern food webs. And as Kohen introduced in the first segment, the amount of oxygen that's been available in the ocean has changed over time, and has changed while eukaryotes have existed. So essentially, when you go back into the past, eukaryote ecosystems were more like the ones on the top. And then the sorts of standard food webs that we're familiar with now, these are recent developments. And of course, this whole webinar series is about ocean deoxygenation. The oceans are continuously becoming deoxygenated. But in the past, it was the inverse, that the oceans were less oxygenated in the past and became more oxygenated. So the past and the present, the past and the future are kind of mirrored in this way. So that sets up like the relevance of this deep time stuff to deoxygenation. And that's a lot of the questions around deoxygenation, or how are ecosystems going to respond to a deoxygenating ocean in the future. A lot of the questions that I investigate deal with, okay, well, how did the earliest eukaryote ecosystems function under a very low oxygen marine biosphere? These are things that many different researchers want to know, and usually different communities represented by people with different backgrounds. But they have something in common, is that the only way to answer these questions fundamentally is to get an understanding of how eukaryote physiology and ecology changes as a function of oxygen availability. Because then, as you know, that how they function at low oxygen, high oxygen, that's the fundamental data you need to answer questions about the past and the future. So I'm just going to give a quick overview of Earth history, like what Kohen did. This is just a timeline. So all the timeline figures, the present is here, 0 billion years ago, and the past is over here on the left. So what Kohen showed is that atmospheric oxygen levels have changed considerably over Earth history, and there's been three main intervals, like essentially vanishingly no or low oxygen, low to no oxygen. Oxygen first became a permanent feature of the atmosphere right around here. So this is like 2.2 billion years ago. Oxygen became part of the atmosphere stably, but it didn't reach modern levels until sometime in the past 500 million years ago or so. So the earliest evidence for eukaryotes from the fossil record, they appear around here, 1.6 billion years ago. But then some of the best evidence that we have for the size or importance of the eukaryotic component of the biosphere comes from fossilized lipid molecules. And we don't get reliable, convincing evidence of these molecular fossils until about 800 million years ago. So we know by then there were definitely modern eukaryotes around, and that they were relatively abundant compared to their abundances in the past. But why this pattern exists is not clear. The idea is that maybe eukaryotes, they were around here, but they were constrained by the environment, and that constraint was lifted around the time these biomarkers enter the rock record. That's the idea. We're going to zoom in on the Proterozoic. That's this interval here. You can just think of it as Earth's middle age. This is just the Proterozoic in focus now. And this is just a bunch of data from the fossil record, the biomarker record, genomic record. Pretty much it's how we know that eukaryotes seem to have been around here, but they weren't that abundant or diverse. And then right around here, around 800 million years ago, the eukaryotic biosphere blossomed, right? And this is the beginning of modern ecosystems, essentially. So I mentioned those biomarkers, these eukaryotic biomarkers appear around then, but then also the diversity in the fossil record increases here. And the earliest definitive evidence we have of like a eukaryotic fossil in a section that is confirmed to be oxic using the sort of proxies that Cohen talked about, that appears around here, so tentative. Okay, so that's just how we know there's this pattern. Eukaryotes are old, but then they diversified 800 million years ago. The amount of oxygen that was available during this time is widely debated, and there's two main possibilities that people argue about. The first one is that oxygen was somewhere between 1 to 10% of present atmospheric levels. So present atmosphere, 100% PAL is what we have now, right? So the idea is that oxygen was at most between 1 and 10% during this interval. And this is coming from the sorts of studies and methods that Cohen talked about. Then there's another scenario that oxygen was actually below 1% during the mid-Proterozoic, and then rose above this threshold, above 1%, around 800 million years ago, in line with this apparent diversification of eukaryotes. So this is a way of setting up the main question is, well, what difference does this make? Like, how would eukaryote ecology function under these levels, 1 to 10 versus less than 1%? Here's an example of like the sort of geochemical data and fossil correlations that define these questions. So like here, this is just one of these redox proxies, right? And there's a shift around 800 million years ago, interpreted to signal a rise of oxygen. And it's correlated with this eukaryote diversification. So this is the best we could do as geologists, is make these correlations. But then if we actually want to know, like, is there a causal relationship between oxygen availability and eukaryote diversity? The only way to do that I know of is by looking at eukaryote diversity today as a function of oxygen, and then having to infer how diversity would have changed in the past as a function of oxygen availability. So this is the background. This is like this is where these questions are coming from. And then to answer them, we essentially need to find like a modern laboratory to look at the effect or control of oxygen concentration on eukaryote diversity. So we did that by looking at a place in British Columbia, off the coast of Vancouver Island. So here it is, zoomed out. There's the US and Canada there in the West Coast. And just zoom in. And then you get this little, this place here called Saanich Inlet. And this is our field site. It's the home of a 10-year plus time series experiment that's looking at the control of dissolved oxygen concentration on mostly the biogeochemistry and ecology of bacteria and archaea, the things I said we're not going to talk about. But people have also, the people who organized this time series have also looked at eukaryotes. So these are the people that I worked with. So we essentially want to go there to test these sorts of geologic questions. So the way Saanich Inlet works, it's consistently anoxic in the bottom water, and that's mostly just due to high productivity. And then also there's this glacial sill at the mouth of the inlet that restricts bottom water circulation and mixing. So those two factors combined cause bottom water anoxia. And here's the data. This is CDT measurements that we made with some fitting to get the intermediate oxygen concentration. So the site that we looked at is about 200 meters deep. And here is a oxygen in micromolar here. These contours correspond with these geologic regimes that we talked about, where like up here is between above 11 to 100% of modern levels. This is 1 to 10, and this is below 1. So you can see how those oxygen concentrations play out in time at this sampling location throughout the year. These were all these measurements were made in 2015. And these are the samples, these are the depths at which we sampled for collecting DNA. And for reference, this is also sulfide concentration. So bottom water, Euxenia also is a feature of Saanich Inlet at certain times of the year. And we've sampled both low oxygen without hydrogen sulfide and then samples with hydrogen sulfide. And essentially we made like 4 different redox regimes to test these geologic hypotheses. So essentially like we just converted, we actually from the CTD have micromolar measurements, and then just using the temperature and salinity at those locations, converted that into a geologically relevant bin. So again, like 11 to 100% of present atmospheric levels. Another way of thinking that is it's the same, it's interchangeable with atmospheric saturation, the modern atmosphere, right? So we made different bins, 1 to 10. Remember that was one of the scenarios for the Proterozoic. When eukaryotes originated, it was around 1 to 10. Then this is less than 1%, right? So that's the other possibility. And then we also have the less than 1% with the hydrogen sulfide as a distinct bin. So we sampled water at these different depths spanning the oxycline, and we filtered the water using 2 filter sizes. So the first one has pores of about 3 microns in length, and the idea is that this is capturing eukaryotes, these larger eukaryotes that are bigger than 3 microns or so. Smaller eukaryotes pass through this filter and then get caught on the second filter. So as a result, we have 2 size fractions. So we're looking at both. So we could look at both the control of oxygen concentration on eukaryote diversity, but also cell size. So we have 2 size fractions and 4 redox regimes. So that's how this data is going to be presented. That's the background. So first of all, for the filters, we just extracted DNA from them, and then we're looking at we use primers that are targeting the V4 region of the 18S rRNA gene, so to look at eukaryote diversity. So this is just a sense of the taxonomy, and this is a lot. The taxonomy as both a function of redox regime and then also the 2 different size fractions. The only thing I want to address here is that this is what you get with large eukaryotes, the large size fraction under like near modern oxygen concentrations. We have the same ordering here. This is the top 10 most abundant major groups of eukaryotes. We have that same ordering in all these other different bins. So you can see that the relative abundances of these different groups are changing as a function of oxygen in temperature, or sorry, in size. So we know that there's an effect there, but this is just to get, you know, and people at Saanich Inlet and at other locations have noted this before, that oxygen does change the community composition. So this is more or less a confirmation of that. But we want to look specifically at diversity. So paleontologists do look at fossil eukaryotes and are able to estimate taxonomic richness, just the like alpha diversity, the number of eukaryotic taxa throughout geologic time as preserved in the rock record. So we could do the same thing. We could use an estimate for alpha diversity or richness using our gene data. So that's what this is. That's what you have here. This is for the large size fraction. This is coverage on the x-axis, but essentially what this says is that we sufficiently sampled, we have sufficient sampling coverage to account for the taxa that we believe are there. So on the y-axis, just think of these as these are operational taxa. They're amplicon sequence variants. It's just an operational designation for microbial taxa. This is what it looks like under the large size fraction, and then the small size fraction, and it's by redox bin. And in line with what other people have demonstrated at places like this, as oxygen decreases, alpha diversity also decreases. But what we're also interested in is how much. So for example, like if you look in the small size fraction, yeah, alpha diversity is lower in 1 to 10%, but it's still fairly similar to what you get under more modern air saturated conditions. But then as you go below 1%, alpha diversity drops a lot. It's a greater discrepancy. And then same thing with Yersinia. Here under the large size fraction, actually Yersinia has more alpha diversity than low oxygen waters without sulfide. A lot to unpack here, but the point is that there is a difference in alpha diversity, and we want to look specifically, and we want to quantify those differences, which I'll do in a bit. But we also want to look at other metrics of diversity. So that also includes phylogenetic diversity. So these different redox regimes, how much of the eukaryotic tree is represented in those samples under those redox regimes. If it has high phylogenetic diversity, that means a lot of the eukaryotic tree is represented. If it is low, that means only certain lineages are being represented. And given these analyses that we did here, there's really only a significant difference that we were able to detect using our tests and our data between the less than 1% PAL bin and then with the modern air saturated bin. Actually, and then that's for the large size fraction. Under the small size fraction, I mean, probably if we got more samples, there'd be we would show a significant difference, but given the samples we have, we didn't recover any significant difference. Then we also looked at evenness, which just shows the distribution of abundances within the community. So high evenness correlates to they have all the different taxa are well represented, respect to one another. And again, we only see a real significant difference between the less than 1% bin and the modern air saturated bin. So let's put this all together and then look at these bins and then try to do this thought experiment where, okay, say in the Proterozoic, the atmosphere did have 1 to 10% PAL, and then it went up to, there's an oxygenation event and it went to 11 to 100. What kind of changes in diversity we would get? Well, at least just using our data as a constraint, you do get an increase in the number of ASVs, 8 to 40, depending on the size fraction. No significant increase in phylogenetic diversity according to our samples. Then if we look at, okay, what if it was less than 1% PL and it rose to 1 to 10? What would that look like? Will there be a greater increase in ASVs? And still no significant increase in phylogenetic diversity. But if we look at this change, then we get a very large, up to 227% increase in ASV numbers. And then we also reflected a significant increase in phylogenetic diversity as you go from less than 1% to 11 to 100. And that's the kind of signal we would expect. You would require something like this, a diversification event like this, to be recorded in the biomarker record, the fossil record, in the genomic record, looking at the tree of eukaryotes and when all the major groups diverge relative to one another. So that seems to be the most, that would be the state change needed to bring about this diversification event. So if we look back to this figure, and I'll wrap this up in a minute, if oxygen really was between 1 and 10% the PAL during the Proterozoic, and it kind of stayed within this range at this time, you would not predict that there would be a diversification event of eukaryotes. If this is true, oxygen was like this and there was a diversification event, something else other than oxygen drove it. Alternatively, if oxygen really was below 1 to 10% PAL, as some of my colleagues believe, and it increased to above 10%, around 800 million years ago, our data showed that would support a diversification event like we see recorded in the fossil and biomarker records. So essentially what we've done for the geologic community is provide constraints for reconstructing how these different scenarios for atmospheric oxygen in Earth's deep past would have impacted eukaryote ecosystems as we know them. So that's essentially the major conclusions. How to tell the difference between these scenarios, people like Kohen need to figure that out. Sequence data in a modern fjord can't tell you that, but it can give you constraints on how to interpret those oxygen levels as people reconstruct them. So thank you, and I'm happy taking questions now.
Okay, thanks very much for a great talk, Dan. We do have some time for questions, so if you have questions for Dan, please populate the question and answers box in the Zoom window. I have a question.
Yep.
Have you thought about how your results might inform our understanding of potential climate or potential impacts from contemporary deoxygenation?
Those are the sorts of things, to be honest, I think mostly about the historic implications, but this is part of the reason I was excited to give this. I would like to see how people interpret that. I mean, I think people already knew that, like, if you go to the cores of OMZs and as these oxygen depleted regions of the ocean grow, diversity is going to suffer. But what I would be interested to see is suffer in which ways, like what kind of, because there's still going to be eukaryotes there, and the eukaryotes under anoxia, what are they doing? And I would like to know that from an Earth history perspective, but also that would be important to understanding how ecosystems are going to change in the near future.
Yeah. So we got a question from Cohen. Dan, do sulfide versus ferrous iron concentrations possibly play a role? During the Neoproterozoic oxygenation event, it appears we may have had intervals of both.
Yeah, so we did look at Euxinia. There's a couple of interesting things about Euxinia. So if we just look at like the large size fraction here, and look at the euxinic bin, it actually does not significantly differ from the high redox bin, but we do know that there are fewer taxa, like fewer ASVs in the sulfidic bin than the high oxygen one. And the way I interpret this is that first food chain figure I showed, anoxic ecosystems aren't going to be able to support as many trophic levels. And so I think numerically, there are going to be fewer cells and alpha diversity will be limited under Euxinia. But what this suggests of phylogenetic diversity is that even though there are fewer taxa there, those taxa represent a broad coverage of the eukaryotic tree, which is interesting in a lot of ways. One way of interpreting that is that adaptation to Euxinia isn't relegated to just a few early branching or odd eukaryotic lineages. It appears to be widespread. And actually that would make sense if eukaryotes originated at a time in Earth history where the ocean was more prone to Euxinia. So I actually, according to this, actually, both the phylogenetic diversity and then the alpha diversity for the large size fraction, these diversity metrics actually look better or more modern under Euxinia than they do under less than 1% PAL with no sulfide. So there's this dogma in historical geobiology that sulfide is the worst thing that could happen to eukaryote ecosystems, especially large ones, but our data don't suggest that at all, which is unexpected and worth looking into.
Thanks, Dan. We've got a question from the audience. Is it from Lisa Levin? Is it possible to map which parts of the modern ocean in 3D will shift from greater than 1% to less than 1% oxygen under current deoxygenation trends? Some areas are likely to experience this.
I hope it's possible. if it is possible, I don't think I'm going to be the one to figure it out or do it anytime soon, but I would really hope so. Again, I wouldn't know how to do that, but I know there are people who are in a position to do that, and I would be very happy if that were possible and done.
Okay, thanks. So I think that brings us to the close of the hour, and that's probably all the time we have for questions now. So thanks again, Dan, for a great talk, and thanks also to Kohen for his talk.
Yeah, thank you.
All right, and so with that, I will wish everyone a pleasant morning, day, or night, depending on where you're from. And thanks again, everyone, for connecting for the webinar.